Welcome to This Week in Infectious Disease. This week we're covering 10 notable papers spanning practical hospital-based infection control, the intricacies of host-virus interactions, and the latest in global health epidemiology and policy. Let's dive in. We begin with a focus on practical prevention and management of common hospital-associated infections. First, a major paper from the Lancet Infectious Diseases addresses the persistent problem of non-ventilator hospital-acquired pneumonia, or NVHAP, in a multi-center stepped wedge cluster randomized trial across 3 Australian hospitals called the HAPPEN trial. Investigators evaluated whether an enhanced oral care intervention could reduce the incidence of NVHAP. The intervention was multifaceted, including enhanced delivery of oral care with specific products, education for both patients and staff, and an audit and feedback loop. Among over 8,800 patients analyzed, NVHAP was confirmed in 78 individuals. The incidence was 1.0% in the control group receiving usual care, but fell to 0.7% in the intervention group. More strikingly, this translated to a cumulative hazard ratio of 0.40, representing a 60% reduction in the risk of developing NVHAP. This trial provides strong, high-quality evidence that a structured and actively promoted oral care program is a powerful and relatively simple tool to prevent one of the most common healthcare-associated infections. Staying in the hospital setting, a new analysis in clinical infectious diseases examines the cost-effectiveness of different approaches to fecal microbiota transplantation for recurrent C. difficile infection. With new commercial, FDA-approved microbiota therapeutics now available, the question of value is paramount. Using a Markov model, researchers compared traditional FMT delivered via colonoscopy to these newer commercial products. The results were clear. Traditional FMT by colonoscopy was the most cost-effective strategy for preventing a second or subsequent C. diff recurrence, with an incremental cost-effectiveness ratio well within the commonly accepted threshold. In fact, assuming a willingness to pay of $100,000 per quality-adjusted life year, commercial microbiota therapeutics were not found to be cost-effective under any circumstance when donor-derived traditional FMT products are available. This finding has significant implications for institutional formulary decisions and practice guidelines, suggesting that at current pricing, the newer commercial products are not an economically viable replacement for traditional FMT if it can be accessed. Next, we'll turn to a group of papers that unravel the complex interactions between hosts, viruses, and the immune system. A report in Science Translational Medicine provides new insights into how SARS-CoV-2 immunity is shaped by vaccination and infection. Researchers analyzed memory B-cell responses in participants who were boosted with either the original Wuhan 1 strain, a variant, or a bivalent vaccine. They found that while variant boosters and breakthrough infections led to a transiently greater recall of cross-reactive memory B-cells, the long-term effects differed. An Omicron booster caused little long-term change in the memory B-cell repertoire. In contrast, an actual Omicron infection resulted in memory B-cells with higher neutralizing capacity against both the original and Omicron strains months later. However, this came at a cost. These potent memory B-cells from infected individuals showed less breadth against more distant variants like BA.2.86. This suggests that while infection can strengthen immunity to recent variants, it may narrow the protective window against future, more divergent strains. Moving to HIV, a highly technical study in Cell used genome-wide CRISPR screens in primary human CD4 plus T cells to systematically identify host factors that either help or hinder HIV infection. This work validated known factors, but more importantly, uncovered several potent new antiviral factors. For example, a protein called PI16 was found to inhibit viral entry, while another called PPID, a relative of the proviral protein SIPA, was found to bind the HIV capsid and reduce its ability to enter the cell nucleus. This kind of fundamental discovery provides a detailed map of the HIV host interaction landscape and identifies novel pathways that could be targeted for future antiretroviral therapies. In a related vein, a commentary in Clinical Infectious Diseases explores the intriguing link between zoster vaccination and a reduced risk of dementia. The author reviews the growing body of observational evidence and proposes 3 potential mechanisms that now require rigorous investigation. First, that vaccination reduces the cumulative burden of varicella-zoster virus reactivation. Second, that it provides a nonspecific beneficial modulation of the aging immune system. And third, that it prevents zoster-associated neurovascular injury that might otherwise contribute to cognitive decline. This piece highlights a shift from asking if there's a connection to asking how it works. Finally, a paper in Nature identifies a specific subset of regulatory CD8 T cells guided by a receptor called GPR15 that are crucial for controlling intestinal inflammation. These cells home to the colon and kill inflammatory macrophages. The study found that deleterious GPR15 gene variants in humans are associated with severe early-onset inflammatory bowel disease, and that these protective cells are reduced in the guts of IBD patients. This discovery offers new insights into organ-specific immune regulation and potential therapeutic targets for IBD. Our final theme covers epidemiology and public health policy. In the Journal of Infectious Diseases, researchers used whole genome sequencing to map the transmission of Mpox in Thailand during the 2023 global health emergency. The molecular epidemiology confirmed that transmission was almost exclusively within communities of men who have sex with men and was dominated by specific viral lineages. The analysis also revealed mutational patterns suggestive of evolution driven by the human ApoBec3 enzyme system. This work underscores the critical role of rapid genomic sequencing and data sharing in understanding and responding to global outbreaks. On the policy front, 2 papers in the Lancet Global Health address key prevention strategies. One makes a strong case for including young adolescents, aged 9 to 14, in the policy development and clinical trials for new tuberculosis vaccines. The authors argue that while there are challenges, such as lower TB incidence in this age group making efficacy trials difficult, the opportunity to vaccinate before the age-related rise in TB risk is too important to ignore. They propose novel trial designs, such as focusing on household contacts, to overcome these hurdles. The second paper from the same journal provides a sweeping 34-year analysis of sugar-sweetened beverage taxes across 183 countries. It found that the adoption of these taxes has accelerated rapidly, now covering 3.5 billion people. Interestingly, multivariable analysis showed that the key predictors of a country adopting a tax were its prevalence of diabetes and obesity and its per capita GDP, not its baseline consumption of sugary drinks. This suggests that policy action is being driven by the visible health and economic consequences of diet-related disease. This focus on global health resolutions is echoed in a health policy paper from The Lancet, which provides a framework for implementing the recent World Health Assembly resolution on kidney health, emphasizing the need for political commitment to improve care for the 850 million people affected worldwide. If you only have time for one paper this week, make it the Happen trial on oral care for pneumonia prevention in The Lancet Infectious Diseases. This multi-center randomized trial provides strong evidence that a structured oral care program can significantly reduce rates of non-ventilator hospital-acquired pneumonia, a common and serious complication. Here are the key takeaways from this week in infectious disease. First, for preventing hospital-acquired pneumonia in non-ventilated patients, an enhanced oral care protocol is a highly effective intervention, reducing the hazard by 60% in a large Australian trial. Second, when considering fecal microbiota transplantation for recurrent C. diff, traditional colonoscopy-delivered FMT remains the cost-effective strategy. Current commercial microbiota therapeutics are not cost-effective alternatives if traditional FMT is available. Third, regarding SARS-CoV-2 immunity, recent data suggests that while breakthrough infection boosts neutralization against recent variants, it may narrow the long-term memory B cell response, potentially reducing protection against more distant future variants. Finally, on a policy level, global adoption of public health interventions like sugar-sweetened beverage taxes is accelerating, driven more by a country's burden of non-communicable diseases and economic capacity than by pre-existing consumption habits. That's your roundup for this week in infectious disease. The full transcript and references are available on the episode page in your Audio Scholar library. This is an AI-curated summary. For clinical decisions, always consult primary sources and current guidelines. See you next week. This is an automated summary generated by artificial intelligence, which can make mistakes. Always review the original source materials.